Hello, and welcome back to the International Journal of Medical Students webinar series, Research Pathways. My name is Dr. Leah Komer, and I'm the host of this series, which features presentations from various editorial members at the IJMS, highlighting their experiences entering and navigating the world of academia, their challenges and triumphs, and their advice to future clinician scientists. Joining us today is Dr. Paul Morgan. Paul has an associate degree in chemistry and biology from the University of Belize and a dual chemistry and biology degree from the University of the Ozarks. He's completed his bachelor's of medicine in Taiwan, and he is currently a physician scientist at the Icahn School of Medicine at Mount Sinai and en route to a PhD in biomedical scientist. Thank you for joining us, Paul, and welcome.
Thank you. Thank you for having me, Leah. Thank you, INJS. Thank you to our audience. It's really an honor and a pleasure to join you guys today. And without further ado, I'll just kind of get started. I grew up in Belmopan City, Belize. I was born in this tiny capital city in Central America and the Caribbean. So here's Belize, and you can see that. I enjoy cake quite a bit. So I think this was my second birthday. So this guy here is me, and I'm sure at this point I was thinking about perhaps how exciting and adventurous this world would be. And as I moved on, and I do reflect on my career and particularly on the research I've done at this time, I would say this look that I do have on my face at 2 years old is still that inquisitive look that I carry in my heart today, as I move forward with with the things I enjoy doing. So I'm also a big sports fanatic. I've done a lot of different sporting activities throughout college and high school, and I'm even trying now at a much older age, but just probably more or less just to stay fit and healthy at this point in time. But I have competed for Oklahoma State University in cycling, and I did track for my undergraduate school. I've done karate and volleyball. I enjoy being active. In fact, health and sports and health and these two things are also connected. We cannot be productive. This is my philosophy: is that we're not ever gonna be at maximum efficiency or be super productive if our mind, body, and spirit aren't connected and aren't healthy and in sync with each other. So my research journey really started maybe about 12 years ago or so. I completed my undergraduate degree, so a bachelor's in chemistry and biology, and I did minors in mathematics and physics at the University of the Ozarks in Clarksville, Arkansas. And at this point in time, I didn't really understand, the rigors of research and so on, but I was extremely curious, and I think that's the first step that we all need to take. We can all be researchers. There's a niche for everybody, and I really put this picture out here as the start of my journey because I was just curious. That's all. I had some basic working knowledge of biology and chemistry, I would say, but my curiosity was, I think, what was the most advanced thing about me at this point in time. And so while I was at the University of the Ozarks, I did different tiny research projects that were connected to my advisors. And in one such project, I looked at gamma radiation on fruit flies and regeneration. Sorry, and survival of fruit flies. And so at this point in time, I had just been understanding how journals and how research and the publication aspect of things are connected. And that is why I'm really grateful for INJS and the work they have been doing. They give young, junior clinicians and scientists an opportunity and a foot in the door. I feel like it's important to also say this because when we apply for, I think about a few years ago, trying to get employment with an undergraduate degree. And one of the things that really discouraged me as I looked for vacancies and looked for opportunities was you need 5 years, 3 years experience, working experience, or everybody asks for 5 or 6 years, 3, 4 years research experience. And I think that we have to take a step back sometimes and realize that we all have to start at zero, and then we move up to one and two and three, and then we can add on things from there. So IMGS, IJMS really gives everybody, enthusiasts in medicine and sciences, an opportunity to take that first step into the world of publications, into the world of the academic rigors of dissemination. Um, Out there. So um, I then moved on to Oklahoma State University. So I worked for 1 year, and at Oklahoma State University was where my curiosity kind of heightened, and I was looking for a niche. I decided I would like to explore beyond chemistry, or something more applicable to biology rather. And there was nothing more exciting to me than structural biology. So this is where I really kind of got into structural biology. I had a great didactic training, fantastic professors, and I had an opportunity to be a part of a research group at Oklahoma State University that really worked on protein structure and function. So this is where all those senses in my excitement and my curiosity really started to take off here. So I think this is one of my seminar presentations here. It's somewhere around circa maybe 2016, 2015, 2014 or 2015, somewhere around that time. But here I'm actually presenting the structure of the CCR5 protein receptor. And this is one of the proteins that HIV uses to enter cells to infect its host. And so at that time and even now, I'm really still passionate about protein structure and function, and it's something that I'm going to talk about in a little bit as well. In 2016, after doing graduate studies at Oklahoma State University, I moved over to Taiwan. I got a full scholarship to pursue medicine. So here I did a post baccalaureate. I enrolled in the post baccalaureate Doctor of Medicine degree, and these were country to country scholarships that were offered from Taiwan to Belize. And so I moved over to a new country, and I learned how to use chopsticks, and I had to learn a new language. And I think my ability to interact with a different in a different culture, in a different environment that was different to what I'm used to, really allowed me to appreciate a lot more things in life, and to appreciate my journey as well. So here I am. I'm very excited. I think this was the first time I really wore scrubs in a sort of a semi-official capacity. I think this was probably what my first rotation, and this guy Alex is still my good friend today. So I spent 4 years in Taiwan, and there I got involved. Well, my first 2 years were very tough. Those were more of the didactic years where I spent, all day 8 to 5 in the classroom for 2 years straight, taking exams and so forth. But towards the last 2 years, I really got more hands-on, more clinical practice, and this was where my rotations really, climaxed. I also got involved in my last 2 years as well. I had more free time to be able to do research and get more involved. And so here, this image to my right is me presenting a poster as part of some work that I had been involved with. In fact, I must say that Ida Hospital is one of the a pretty progressive hospital in Taiwan. It's a private institution, and they are very deeply connected to research. In fact, you can cross over from the hospital into the research institute. And this was really how the IDA medical healthcare system was really designed. So again, my curiosity took me, between the clinic to the research labs, and I spent a few months after I completed my MD. I spent a few months also doing research in the Department of Gastroenterology and Hepatology, and there I worked on mouse models to try to replicate HCC and to try to define and basically elucidate some of the pathways in cholangiocarcinoma and hepatocellular carcinoma. So there I also worked on structural biology as well, but this time I kind of transitioned over totally to computational structural biology. So I looked at mutations and how point mutations perturb protein structure and function. So here I am actually presenting at one of the I think conferences at some point. I think this was an imaging and therapeutic conference, something like that. But I was pretty excited about presenting some of this independent project that I was actually working on. So I graduated, and I was really excited about that. I worked for 1 year at home. I tried to basically enroll in the internship program in Belize, but that was just for a year, and I saw that as an opportunity to transition into something bigger career wise. And so I landed myself in New York City, New York, in the heart of the concrete jungle, and that's where I am now. So here I'm actually pursuing a PhD in biomedical sciences, and I get to really work on an assortment of things, but I'm actually tied to the Department of Nephrology. So I'll talk a little bit about that in a minute. But here I was really excited about the opportunities that Mount Sinai offers. So Mount Sinai is not exactly a university, and that really was my intention. My intention was not to really go back to a university to pursue any form of training, but rather I wanted to enroll in a degree granting hospital, and so Mount Sinai really stood out as one of those institutions where I would be able to still immerse myself in academic medicine as I prepare for my next step, which would be the board exam. Um, and perhaps still, getting acquiring the PhD that I've always wanted to get. In fact, as far as I know it, there are no Belizeans yet. There's no Belizean yet who has completed an MD and a PhD. So hopefully, I'm still on the path to being the first Belizean to do so. And that is because I'm really proud of this journey. Not that it's totally uncommon to have an MD and a PhD, but I think that it represents the heart of what medical research is about and how we can actually advance medical research. And if you think about a country that I come from, a beautiful country in the Caribbean, but also a resource, stricken environment where we don't really value, research. I think that most of that can be attributed to maybe overall ignorance in terms of how important research is to advance our hospitals, to advance our healthcare system, and so on. So these are some of the things that I would like to pay attention to after I complete, some of this training that I'm doing here, is to really use these experiences and this wealth of knowledge to uplift my home country and my community. And there's a long ways that we need to go. In fact, I'm yet to see that we have a national health insurance, and this is something I was exposed to in Taiwan. I'm yet to see that all our healthcare facilities have basic things like a CT scanner. I'm yet to see that we also embrace our youth who have had to step out beyond our country's borders to get, basic medical degree and basic training in medical sciences and biomedical sciences. So we're doing better, but there's a whole lot more that I would like us to do, and I'd like to bid to continue to being part of this process. I must say also that I also teach at the University of Belize as a very part time adjunct. So I mean, I think attests to some of the passion I have to give back to the youth to share the knowledge that has been given to me. So I do basically mentor undergraduate students pursuing their bachelor degree in thesis, in their research thesis. That's their capstone project. So I don't know, I no longer teach courses anymore, but rather I mentor in terms of research, and I kind of handle a few students every year in with their capstone projects, and particularly in biomedical sciences and pharmaceutical sciences. Um, so now I get into what I really do now, and what I really do here as part of my PhD training is to work on rational drug design and development. So this is really the cornerstone of what I do, and rational drug design and development um, basically uh, can be broken down into two major approaches. The first is what we call a structure based approach. So, a structure based approach is where we can take large libraries of small molecule, potential small molecule drugs or tiny peptides, and we can basically dock these peptides and screen these peptides against pathologic protein targets. So, if there's a protein target that has been implicated in epilepsy or in COVID or in hepatocellular carcinoma or in epilepsy, we can target these proteins. And so I use large libraries, large virtual screening libraries, and combinatorial libraries to really look at how we can effectively drug targets. The second approach is a more kind of chemistry based approach. It's a ligand based approach that I also really have a great deal of experience in. And this ligand-based approach is basically where we take a known drug, maybe let's say a drug that like for instance, an HIV drug like amprenavir. We can take this drug and we can improve it. We can try to maybe look at a different target or evolve it into some new molecule with less toxicity and maybe better efficiency. So that's what basically a ligand-based approach is, where we have some structural information, some target, some inhibitor, or some activator for that target, and we evolve it into something new or different or better for another class of protein families to treat a particular disease. So personally, I'm more excited about the structure-based approach. That's really where you really discover like novel things. But both has its place. Um, I've also used some of this knowledge to, be involved in things like protein engineering and de novo protein design. So these are some of the things that I work on as well. In the first 2 points, you'll see that sometimes small molecules aren't the solution to all the problems we see with certain diseases. In fact, one of the major pharmaceutical drug. The best-selling pharmaceutical drug in the world is Humira, and Humira is actually a protein drug. It's not a small molecule, right? It's a protein biologic, an antibody. And so sometimes small molecules aren't the answer, and this is why we turn to things like peptides and protein engineering. And to heighten the curiosity, de novo is really building proteins with from scratch without any known scaffolds to do some particular therapeutic function. So most of my time really I spend in the department of nephrology, and part of my work really involves developing new therapeutics to treat kidney disease, and in particular, I work on FSGS, focal segmental glomerular sclerosis, in the department of nephrology. And currently, there are no podocyte, which is the fundamental cell for the kidney filtering. Currently, there are no podocyte specific drugs. So we're really trying to make inroads here into developing the first in class um, podocyte specific drug. Um, so if I get into a little bit more specifics about some of the work I do, um, I, I would say that currently three of my main targets involve UPA. So UPA is one of the main targets that I look at, and UPA is basically a short um, uh, term for urokinase plasminogen activator, and basically UPA is involved in many pathologic processes like FSGS, and it does so by producing lots of proteins called plasminogen, and plasminogen is involved in a lot of in good things in the body, but it's also involved in things like tumor remodeling, and it's also involved in processes like damage to podocytes as well. So UPA is one of those targets that I look at. And one of the other targets I look at is UPAR. That's I've recently picked up UPAR. So UPAR is the activator for UPA, and you can't exactly look at one without looking at the other. From a global perspective on this therapeutic strategy we're trying to employ. And I've also looked at integrin receptors here, and in particular, I've looked at the integrin AV beta 3. So these three proteins, UPA, UPAR, and integrins, are involved in kidney disease and in damage to podocytes, and they're also involved in tumor metastasis and particular tissue remodeling. But for the purposes of this presentation today, I'll just briefly discuss one of the projects that I work on in terms of tumor metastasis, and the second one that I've done previously on using these rational drug design techniques to answer questions in terms of the COVID pandemic. But I mean, now we know a lot more. But at the time I worked on this, we really were working with limited information, and we were really trying to add to the body of knowledge here. So one of the first, I'll talk about two papers, but this is the first one that I'd like to talk about, which is the structure-based discovery of a novel class of small molecule pure antagonists of AV beta 3. So in this work, this has been a long-standing collaboration with Memorial Sloan Kettering Hospital, particularly the Rockefeller University, in conjunction with Mount Sinai, or the Icahn School of Medicine. And this has been a long-standing project that I was just a part on for a year or so. And basically, what we did in this project, just a very brief summary, but what we did in this project was we basically developed a new small molecule here in science. And this small molecule was able to inhibit this very important protein, AV beta 3. And I just want to reiterate that AV beta 3 is actually a critical integrin in tumor metastasis, and also in kidney injury and podocytopathies. So essentially, what we found was that we started basically with a large scale virtual screen where we looked at 2 billion compounds, and we really filtered down these 2 billion compounds to like about 10 to 15 promising compounds. Um, we used those promising compounds and we did some chemistry engineering on them, and we basically developed a very novel small molecule that was able to inhibit this AV beta 3 integrin. But one other thing I want to talk about integrin is that integrin um, is a complex protein, membrane protein, and integrin can adopt many shapes. So proteins are beautiful, and that's why I fell in love with them, because they can adapt many shapes. And each shape is specific to some particular function of this protein. So integrin and this particular AV beta 3 has three shapes, I would say, or three morphs. The first is what is called the compact close, which is here. Um, Depicted here. The second is called close extended, which is here. And the third form is called an extended open. And so these are actually experimental images. So we use state of the art cryo EM at the New York Cryo Center here in New York City. And basically, we can look at this protein and observe which form it's in. Now, why are these forms important? It turns out that the closed compact form of the AV beta 3 is the form that's benign. So when the protein is in that form, it's inactive, it doesn't cause trouble, it doesn't allow metastasis, it doesn't allow remodeling of tissues. When it's kind of in the open extended form, sometimes it is triggered into the extended, from the extended closed conformation here to the extended open conformation. And this is where all the trouble happens. So here is where it really gets involved in tissue remodeling and metastasis. And in fact, in 2022, we know that really the worst thing about cancer is not that we cannot develop therapeutics or antibodies or peptides to really control the replication or angiogenesis, but the problem, the biggest problem in cancer, I argue, based on an assimilation on an assimilation of experts, I would say is really the metastasis. That's what kills patients. So here, this inhibitor really works on trying to curtail and eliminate the ability for tumors to metastasize. And so locking it into this closed compact conformation really inhibits this the ability for these proteins to metastasize. So if we look at some of the results from this study that was published, we see that um, sil- silengitide, which is actually an anti-cancer drug, particularly an anti-metastatic drug, which is in phase 2 clinical trials, was outperformed by our compound, which is MSR, Mount Sinai zero 03, because our compound was able to actually keep this integrin mostly in the closed compact form, whereas silengitide really kept it, mostly in the extended closed form. So we have shown proof of concept here that we can actually develop compounds that can have specific biological activity, specific phenotypic activity on these integrin receptors. So we're really excited about where this project is going. And again, this can take a direction as I'm really envisioning in terms of first in class drugs to treat podocytopathies. The last and final thing I would like to talk about here, a project, was something that really, had was I was really passionate about. And again, I look back at that picture of my second birthday, and I had those curious eyes. And when the COVID pandemic started, I was actually in my last semester in medical school, and can recall it was around February that February or March, they solved part of the crystal structure. They solved the protein structure of the spike, the COVID spike protein. And that really excited me because I thought, wow, that's where a lot of therapeutic potential will come from. And of course, this was a time before vaccines, before even any drugs were developed against COVID. And so I quickly mobilized resources, and I jumped on the opportunity to see if we could use this newly published crystal structure of the SARS-CoV-2 spike protein. And, if we could see where this would go. It turns out that that project I was working on was a little slower than the rest of the world, obviously because of resources in every way possible. But I did get some really interesting work out of the COVID project that I had actually spearheaded. And I decided to take an angle from the first drug that was reported. So this was a large scale clinical study that was done in the UK, and they elucidated dexamethasone as really the first drug to treat, COVID-19, the first effective drug at treating COVID-19. Now, today we have a lot of things, vaccines, all kinds of drugs. Pfizer, Merck, many companies have now put things on the shelf to treat COVID. In fact, I've really understood that now they're using de novo protein engineering to develop inhaler vaccine inhalers or de novo pet proteins to really block the spike of the protein and through inhalers to inhibit infection. So there's a lot of cool things, in town, and I think this work is not so exciting anymore, but I think it does represent our ability to mobilize quickly as a global community and using our knowledge and collaborating, to develop really, I would say breakthrough understandings of diseases. And if nothing else, the pandemic taught us this. So in this work, I really looked at answering the question, part of the question, as to maybe how dexamethasone does work. So we know dexamethasone really works at the transcriptional level, and does, a lot of, genetics and epigenetic control of viral replication, and not, I'm sorry, of immune system responses. But what I suspected at the time was that the report from the UK said that, it couldn't account for really the entire efficacy of dexamethasone. But at that time, it was unknown as to, how dexamethasone really worked in treating COVID. So I threw out a wild hypothesis after talking to a few collaborators and my mentor in Taiwan, and I said that perhaps I think that dexamethasone may have some direct interaction with the COVID virus, some protein. And at this time, more and more proteins were being solved, right? So structures of the proteins were being solved. And as you know, that's exciting for a structural biologist like me when a protein, when a structure is solved. Because I then put on my pharmacologic, my drug development cap, and we can really drug these proteins and use them as therapeutic targets. So I put out a hypothesis here that I think there may be some interactions with not just the COVID virus, but maybe actually a direct interaction as well with our immune system proteins, which would be our cytokines and chemokines. And as you know, one of the major culprits for inflammation and is interleukin 6. In fact, interleukin 6 and TNF alpha are very much related to age-related diseases. And one of the major factors that drive age-related diseases is actually inflammation, right? So Alzheimer's, one of the drivers for Alzheimer's, is actually inflammation. Um, and we see that in terms of heart disease, we see that in terms of many, many, many other pathologies. So basically, through computational work, I proposed that dexamethasone might actually be able to inhibit a few of these proteins. And so that was what the study was about. So I used a host of computational methods, virtual screening, molecular dynamic simulation, and use and metadynamic inferences. And what I found was that indeed, at least preliminarily, we see direct neutralization of many of our hyperinflammatory markers, so our interleukin 33 receptor, our interleukin INFGR, we see interleukin 21, as well as key players. So these cytokines and chemokines also have a backstory, and the backstory about these cytokines and chemokines is that they actually seem to be overexpressed in patients who have severe COVID 19. So some of us really can handle the COVID very well, but there are at least a group of um, people for unknown reasons who would have these cytokine storms, these major flare-ups. So the immune system goes berserk, and so basically neutralizing or blocking these cytokines, I suggest that would have some beneficial therapeutic effect. The last thing I would like to mention is that basically what this study represented, although it's much older now, is that drugs in general, first in class drugs, the mechanism isn't really known. We just see the therapeutic effect, we just see the phenotypic effect, but it is long after, it is years after a drug has been shown to have or some small molecule or some protein has been shown to be therapeutically effective. It is usually then we start to understand more about the mechanism. So this is a classic example of trying to understand more about dexamethasone for treating a new disease like COVID-19 at that point in time. So it's one of the classic concepts of phenotypic drug discovery. And this arches or this rivals what you call target-based drug discovery, where we really look at proteins and targets, and then we try to drug them and see if they have some phenotypic effect. So these two strategies are kind of rival, more or less work together, but they somehow conceptually rival each other in the academic setting. So I would say that the skills I've developed is, has been very versatile. We can look at, they look at multiple approaches and multiple strategies. Um, and hopefully in the most unbiased ways possible. Um, Finally, I also worked on other things like epidemiology. So I was also curious. Again, that curiosity had me looking at things like using mathematical models, sorry, Markov models to really predict things like phantom cases at points in time. And so basically, it was a public health approach. And so I said, well, I can definitely use some of the things I'm curious about, Markov's models, and some physics-based approaches to and modeling to really understand how COVID was, really, you know, cycling through our communities. So one other thing that's important to understand is at this time I was doing the work, we hadn't really understood. There was a big buzzword which is asymptomatic cases, or these phantom cases. So a lot of people were infected and didn't have symptoms. So there were a lot of buzz and talks about different things. And of course, my country, Belize, which is very much developing, we're always slow at getting testing and getting the antibodies, getting drugs, against COVID. So we were really slow and behind the first world. And so basically, this mathematical modeling project really allowed us to use these modeling and to make predictions and to make better inferences about how the waves and how COVID really cycles through our population at that point in time. But as I say, all of this is kind of obsolete, or sort of now, because we've really understood COVID much better. We have therapeutics against COVID, and so we're no longer in the dark ages of 2019 and 2020. So with that, I would like to end my presentation, and I'm really, really grateful for this opportunity to share my journey with you guys. Thank you, Leah.
No, thank you so much, Paul. That was an amazing presentation. You have such like diverse experiences having done training all across the world and really putting that emphasis on like scientists within that clinician scientist role, like bringing things back to, those basic sciences, the biochemistry, all those various pathways that as medical students we roll our eyes looking at, but they really are important in terms of, developing these different treatments and understandings of various diseases. And it's incredible the work that you've done in the field of nephrology and even COVID-19, and these public health and epidemiology pursuits that you have too. Quite incredible work. And so that kind of leads into my first question for you. Like how has your biomedical research sort of helped your career thus far? And do you think that there should be more of an emphasis on the biomedical sciences in medical school?
Um, that's a fantastic question. I mean, I think it's packed, and I'll try to see if I could really condense my answer. And this one is very passionate and dear to my heart. So, um, coming from a developing country like Belize, and I love my home country, but we were so naive to how important biomedical research is, and just in general, I'd say research overall, and the impact it really has on our healthcare system and the quality of the physicians and scientists we put out there. So it was until I really got exposed to training in the United States and in Asia, in Taiwan, that I really sought to really appreciate the importance of biomedical research. And in fact, medicine, I argue, doesn't move forward without these advances in research. And of course, I just mentioned research broadly because Without the translational research, the basic science research means nothing. Without the clinical research, the translational and basic science research means nothing. So when you put these three major elements together, it just represents research, biomedical research, and they all go hand in hand, and it's really important for the overall picture as we think about treating diseases and different conditions that we see. So I think that um, I've seen as well here, although I didn't do my MD at Mount Sinai. I do see that there's an emphasis and encouragement to offer physicians in training a gap year, or a year of scholarly year, they call it here, where they take a year gap between their third and fourth year, as they prepare to sit the step two, to do research in whatever specialty they are intending to apply to or pursue. And so it's highly encouraged. And in fact, matching into certain specialties is almost impossible without doing the scholarly year. In many, particularly in fields like dermatology, um, matching into residency is much more promising, taking a scholarly year, which is a dedicated year out to do research. I've also seen that Mount Sinai has a lot of MD, PhD students as well. So this is like 8 year journey. I mean, it's no light undertaking, and it's a major commitment that you should be mentally prepared to undertake. But I think it really shows the dedication and commitment to medicine and sciences overall. But here, we're full of MD PhD students, and they're really on this journey to really push the boundaries and advance medicine. Of course, I must say that you don't necessarily. I'll be honest, you don't really need a PhD if you're curious, you're passionate, and you've completed your medical training. They're hand in hand, and you should have the knowledge just as a PhD. So it's not entirely necessary, but I think the philosophy of pushing the barriers of research is what is most important. I like my global perspective because Taiwan is also trying to push towards giving opportunities to pursue more MD PhDs studies as well. So they're a society that's used to the Bachelor of Medicine, and then Bachelor of Medicine, Bachelor of Surgery, and then maybe a master's degree and specialty, or maybe not even a master's degree, just a Bachelor of Medicine, Bachelor of Surgery, and then on to residency training. But now they're really trying to evolve their medical system to incorporate this MD PhD direction, and they really do understand how important research is to medicine. So I know I said a lot, but I think that that's the only way we'll advance. Being passionate about research and not necessarily getting another degree. Doesn't have to.
Yeah, the extra letters are a nice touch, but maybe not necessary. Um,
Um,
no, so that's excellent that there's those opportunities out there for people who certainly want to pursue it, and it might be a longer road, but large payoffs at the end of it. And so you mentioned a lot about your global training experience and the influence that that's had. And you had some like really great visions of how to improve medicine even like within your home country and other nations as well. Do you hope to go into more of like a public policy sort of role, or how can you use your role as a clinician scientist to advocate for changes in health?
that's a fantastic question. And again, my curiosity leads me to things like health economics and policy. I had some training through Harvard, as well, the Harvard's public health college, as well. In Japan, I did some of this training, um, and they had really brought a team over, but and I kind of got a scholarship and that's where I really stepped my foot in deeply into the world of policy and health economics. In fact, my undergraduate, I also have a minor in economics, so economics and health started to cross, and a lot of things started to really click in my head. And I would say that I really thought at first that being a consultant specialist is the way to change the field, to educate the public. But I argue that yes, it is very important to become consultant and chair and head of a particular department in the hospital. But even more important, we reach people through policy, and that's where things happen. That's where the magic and excitement and changes take place. It's at the policy level, and not necessarily at the not entirely at the micro level, but there must be some macro approach to it. And I say this because in my home country, in Belize, you do not necessarily have to have any training in public health or in medicine, any at all, to be the minister of health. But at the same time, you as the minister of health or the head of health state is the one who makes major decisions and important decisions about pandemics and about distribution of drugs in the country and which facilities should get what kind of attention. And so I really want to reiterate the point here that things really happen at the policy level. So at some point in time, if you're going to make changes, if you're going to be a part of change in your healthcare system and change for the better, there some part of your foot would have to be in policy making. And so I think that would be an inevitable step for me in the future.
Certainly, it's really highlights those like areas of advocacy more like upstream and the policy level, and also the importance of sort of like those downstream advocacy, even at the patient level too, and how as clinician scientists maybe we have a role to do both.
Absolutely.
Yeah.
Absolutely.
And so, I've really liked how, again, how passionate you are about the field of research that you're doing. And you had mentioned that, back when you were in university, you had, great professors and a great research team, which really helped drive that passion for you. And in your opinion, what makes somebody a really great mentor, for somebody who might be interested in research?
So I think great mentors are born, and all of us were born, so we can all be great mentors. That's the caveat there. But it's one who really sees no barriers at all. And I always think about an infant, a neonate. So when I rotated in the pediatrics department, I think about I used to count in my head how many times the mother would tell the child no at the store or in the hospital. And a child hears no like almost 400 times a day. And so no's represent barriers, no's represent obstacles, no's represent why this shouldn't happen, why that shouldn't happen. A great mentor sees no barriers, and it's free, right? It costs us nothing to see zero barriers in front of us. Of course, we have to be realistic, I'd say, but I think the passion is free and it comes from within us, and we should all be mentors. We can all be. In fact, I learned very early on that if you don't know what you want to do with your life, some want to be doctor, nurse, lawyers. If you don't know what you want to be in life, be a teacher. And doctors are teachers, lawyers teach law students, so we can all be teachers. And without great teachers, without teachers that show care, regardless of color, race, identity, social class, teachers that are passionate about giving back, about sharing their knowledge with others. Without that, we really wouldn't advance as a nation, as as global citizens, as citizens of this world. So I would say that it costs nothing to be a fantastic mentor. All it takes is to see beyond obstacles and to try to be a part of others' lives.
Certainly. And my last question, what are some of your future plans?
Well, that's a tough question as well. So I'm about halfway through my PhD now. Rolling on downhill towards the end. Um, I do see myself, still trying to complete, my residency. That's something that's really urgent on my mind. However, I do have a wild card I must share with you finally, and that's my considerations about going into industry. New York, the New York, New England area, they're filled with lots of pharmaceutical companies and biotech companies, and I think lots of my curiosity as a physician scientist might actually lead me to a very different career than a typical physician. And I think my journey, as I wrap up this statement, I think that my journey has been a test has been atypical. It's been a very different path than most other people, and so I really embrace the uncertainty and even maybe a slightly different direction than initially I thought I would be. So I'm quite comfortable with not knowing the exact answer, but we'll see how it goes from here.
Certainly. And yeah, you're totally right. Like it's embracing that level of uncertainty and you know, going into medical school, I think a lot of people think, you go to school, then you become a doctor, you go into whatever specialty, maybe you're a surgeon, all these different things. But there's a whole other side of medicine as well that you can use those skills for. And I'm sure you're going to end up being an excellent mentor to someone who might be more curious about those other options.
Absolutely.
All right. Well, thank you so much again for your time, Paul. We really appreciate it. And I would like to thank our audience as well for tuning in and hope to see you at the next session.
Thank you as well, Dr. Komer. Thank you for having me here. Thank you to IJMS. I think the work that we're doing is fantastic. We're growing, and I'm really just excited about where things will go from here. Thank you so much.
Oh, you're very welcome. All right. Take care.